are set to hear from our third speaker this morning. Our third speaker is Dr. Haribubu Arthanari. We call him, and most people just call him Hari. Hari is an assistant professor in the biological chemistry and molecular pharmacology department within the Harvard Medical School and the Dana-Farber Cancer Institute. Today, he will be talking with us, and the title of his talk is Leveraging Tailored Isotope Labeling and Novel Pulse Design to Encode Amino Acid Selective Line Shapes. So let's welcome Hari.
Thank you, Kevin. I hope you guys can see me and hear me talk.
Yes, we can.
Thank you. First of all, I'd like to thank Kevin and the organizers for the opportunity, and more in particular, scheduling me ahead of Lewis, who normally sets the bar so high. If I had to follow, my title of the talk would be Why Bother? So as Kevin alluded earlier on today, the use of isotopes is pivotal in NMR investigations. Other than making our nuclei visible, the two common uses of isotope is one is to reduce the complexity. Here's an HSQC spectra, and if I could wave the magic wand and color the amino acids, I can use amino acid specific labeling to reduce the complexity of the spectra. And the second more common application is to aid in relaxation. As you know, when you substitute a hydrogen to a deuterium, the gyromagnetic ratio of the deuterium is 1/6, which enters the relaxation equation in a quadratic manner. So you can see that how this would aid your relaxation properties of the observed nuclei. Today, what I want to share with you guys is the use of isotopic labeling to enable amino acid specific line shapes, which is a kind of an uncommon way of looking at labeling. And the work was done by Scott Robson on the biochemical side, and Paul Coote on the pulse design side. But just before I go into the details of the talk, let's just set the stage. All of us know that this fingerprint spectra, the HSQC, is a very powerful spectra that helps us look at interactions, look at dynamics, and etc. And it normally takes about anywhere from about 5 minutes to an hour to record such a spectra. But the bottleneck of using this powerful spectra is our ability to correlate back each one of those dots to an amino acid in the primary sequence, often referred to as the assignment procedure. This is normally traditionally done using a set of triple resonance experiments where we're able to transfer magnetization along the backbone of the protein and taking advantage of the fact that the nitrogen is coupled both to its own C alpha and the C alpha proceeding to it. Now, so what's the problem? So here are two spectras, one of an 8 kilodalton protein, another of an 80 kilodalton protein. And what you would immediately see is there, and then when you go to an 80 kilodalton protein, there are more resonances. It becomes crowded. And secondly, the 80 kilodalton protein also relaxes much faster. Now, more resonances translates into your assignment problem, something like this, where, for example, if I'm sitting on an HNCA looking for a match for my CA-1, there are multiple peaks that raise their hand up and say, I would be a potential match. Now, the second problem of the relaxation comes in your sensitivity of the triple resonance experiment. Here's a slide from an old review by Michael Sattler, which still holds good. Here's the relative sensitivity of the different triple resonance experiment. And you can see that the HNCA experiment is the most sensitive experiment that gives you sequential connectivity. And the sensitivity of the HNCA could also be debated in high field magnets where the CSA of the carbonyl would be a problem. Now, to put this more into a relevant perspective, here is a phosphatase that we were working on. Here's a TROSY HSQC on your left-hand side, the projection along the HNCA, where we see the majority of the peaks, and then you see the HNCA-CB, you see a very small fraction of the peaks. Now, this renders the HNCA to be the most sensitive experiment to do our sequential assignments. But the problem in using just the HNCA alone as the experiment to do the sequential assignments is the degeneracy in the C alpha chemical shifts. So here I plot the C alpha chemical shift from the BMRB database as a function of different amino acids. And you can see that most of the amino acids, the C alpha is centered around 55, with the exception of glycines and some other prolines that are slightly downshifted. And this is why we use the C beta chemical shift, because the C beta chemical shift has a larger dispersion. And we use that to disambiguate any degeneracies that occur in the HNCA. But for larger proteins, HNCA CB seems like a want. So we started off with a very simple question, is that can we extend the resolution in the C alpha dimension of an HNCA spectrum, thereby mitigating some of the problems that happen with degeneracy? Now, how do we extend the resolution? It's pretty simple. You just acquire the FID for a little longer. So you get 3 different FIDs. And then, as you can see, as you acquire them for longer and longer, you get better and better resolution. But this statement only holds good if your FID is not affected by relaxation. And here you can see that the same FID now relaxes to 2.5 times faster. And it doesn't matter how long you acquire the FID for, you're not going to gain any resolution. But fortunately for us, when a protein is deuterated, the C alpha is one of the slower relaxing nucleus. So here I show you the relaxation rate on the y-axis with the molecular correlation time, or the molecular weight of the system on the x-axis. And here you can see that, let me turn on my, my pointer. Here you can see that the C alpha, when it's protonated, but as compared to the C alpha it's deuterated, when it's deuterated, the C alpha relaxes extremely slowly. It actually even relaxes slower than your the TROSY component of your amide proton. So we thought we'll leverage the slow relaxing C alpha to mitigate some of the problems of a degeneracy. So theoretically, you can take a protein that Lewis works on, like the proteasome, and if you acquire your spectrum in the indirect carbon dimension to about pi T2, you can get a resolution of about 14 Hertz in the C alpha dimension. All is good. If you now believe me and stop the talk and go and record your spectrum, you'll be there up for a disappointment in the regular paradigm. Because what happens is that the C alpha is also connected to the C beta, and your resolution will be limited to about 35 Hertz, where you'll start seeing the homonuclear coupling to the C beta. Of course, this is one of the older problems in NMR, and we thought we have enough tricks in the book to solve this problem. One of the things you can attempt is do a constant time evolution of the C alpha. But for the resolution that we are talking about in about like single Hertz, you need to acquire up to 3 or 4 constant times, about 90 to 100 milliseconds of evolution. And there you can see that even for a very small protein like GB1, you can see that you gain resolution, but you lose intensity. And you lose, and this is for about 3 constant times. And this wouldn't even work for GB1. So for ubiquitin, if you want to scale up. So constant time approach is not a valuable solution. You can use the traditional out-of-the-box C beta decoupling pulses, and these have 2 problems. One is that they cause Bloch-Seeger shifts unless you use them twice, and they also affect the residues at the edge, which are the glycines as well as the serines and threonines, and this didn't seem to be a valuable solution for us. The third option we considered was virtual decoupling. Knowing what the coupling is, you can deconvolve during processing. So here I show you on the left-hand side a coupled spectra. It's essentially a 3D, and you're seeing a strip, and you can see the homonuclear coupling to the C beta. Now, if I assume the coupling is about 35 Hertz and deconvolve, I can deconvolve one of the peak, whereas the other peak gets distorted because the coupling here is not 35, it's 38 Hertz. Now, if I assume the coupling is 38 Hertz, I can deconvolve this, but then the one with the 35 Hertz gets disturbed. So none of the 3 tricks that were on the book were optimal for us. So we naively thought, and we were so stupid in thinking about this, but it turns out at the hindsight, we found something interesting, that we could remove the coupling to the C beta biochemically, ideally, that is making a protein that is only labeled carbon 13 at the C alpha position, and it's not labeled at the C beta position. And in order to achieve this, we thought we'll use pyruvate as a carbon source. Pyruvate is a 3-carbon system, and you can go to CIL and get this labeled at either of the positions, carbon 1 labeled at carbon 2 or carbon 3 position. You can also get the 2,3 labeling, and I'm pretty sure Kevin can correct me. I think you can also get the 1,3 labeling C13 labeled pyruvate. Now, if you use pyruvate as the carbon source, now let's see how alanine gets synthesized by a simple aminotransferase. Now, if I use 2C13 pyruvate, I would get a C alpha that is labeled and a C beta that is not labeled. Brilliant. But the brilliance stops right over there, because the other amino acids are synthesized by some complex mechanism, either using the TCA cycle, the gluconeogenesis, or a direct mechanism. So depending on the cycle that the amino acid gets derived from, the labeling pattern of the C alpha would vary. So now here is, you can go to some of the standard databases and you can map where the C alpha would end up, depending upon the source of pyruvate, the labeling pattern of pyruvate you choose from. Now, things that, for example, if you take things that comes from alpha ketoglutarate from the TCA cycle, will predominantly get the C alpha incorporated from the third position of the pyruvate. But things comes from oxaloacetate will get it from the second position. Things from the gluconeogenesis pathway will also come from the second position. If I would have first referred to this before doing the experiment, we would have quit right here. But we were naively stupid, and then we said, okay, let's do the experiment. But there's one more complication in the amino acid that get derived from the TCA cycle. That is, pyruvate enters the TCA cycle as a 3-carbon moiety and then fuses to form a 6-carbon citrate. Now, this fusion will reintroduce the coupling between the 2 carbons. So you'll have a coupling that is reintroduced. So this was a complete mess, but we were naively ignorant of it. And we made 3 samples. One is the sample with 2,3 pyruvate, one a sample with 2 pyruvate, another sample with 3 pyruvate. Here you can see with the 2,3 pyruvate, you can see again, this is an HNCA, and you're seeing a strip. And you can see the nice coupling to the C beta, both for alanine and glutamate. And now if you take the 2 pyruvate, we already saw that the C alpha of alanine comes from position 2, which gives us a beautiful singlet. Whereas if you go to the glutamate, we saw it comes from position number 3, hence the glutamate's intensity is vastly reduced. Now, if you grow a sample with the pyruvate labeled at position 3, alanine, which derives at position from position 2, is diminished. And glutamate has a very interesting pattern. It has a singlet and a doublet. The doublet is introduced by the coupling in the TCA cycle. So none of these are optimal for us to do an assignment. So what we decided to do was, to cut the long story short, we decided to use a mix of 2 and 3 pyruvate together and grow the bacteria. The idea here is that when the recoupling happens during the in the TCA cycle, the odds of getting the carbon-carbon, C13 labeled carbon, 2 carbons next to each other is diluted. And when we do such an approach, we get this very interesting spectra. Alanine is a beautiful singlet, devoid of any coupling to the C beta, whereas glutamate is has 2 components. One is a singlet that's nice and prominent, and then it has this residual coupling that comes from the amino acids combining in the TCA cycle. Now, what's more interesting about this phenomenon is that every single amino acid has an interesting peak shape. Now, you can see that for alanine, it's a perfect singlet, similar to glycine, which doesn't have a C beta. But for all the amino acid, you have a singlet, as well you have a shoulder. And this shoulder depends on the level of the shoulder, which comes from the coupling to the C beta, depends on the biochemical pathway where this amino acid is derived from. So essentially, you have a central peak that you can use to align, and you have the shoulder, the residual shoulder, that gives you amino acid specific information. Now, you can take the ratio of what we call the uncoupled to the coupled resonances, and then you can plot it for different amino acids derived to a different pathway. And you can see that the best ratio, the worst case is about 50%, which means your central peak is going to be almost twice as tall in almost all the amino acids. And these shoulders give you different ratios that can be used for assignment procedures, which I'll show you in just a second. So again, just to recap, you have a central peak that is devoid of couplings, where the neighboring carbon is not labeled. And then you have the shoulder that depends on the specific pathway. Now, here is one example of how we use this enhanced resolution to aid our spectral assignment procedure. Here is a regular sample made in glucose. And here we acquire it on the carbon dimension to a normal resolution of about 40 hertz, which is what commonly is done. Here we take the same glucose sample and increase the resolution to about 4 hertz. Now here you can see that we have a perfect overlap of the C alpha minus 1, shown in red and green. And in this case, we are not able to distinguish them because they're sitting right on top of each other. Now, if you take the pyruvate premix sample, you clearly see the C alpha and the C alpha minus 1 peak because these are devoid of the C beta coupling. Now, let's do an exercise over here. So we're going to take the protein MBP, which is about 40 kilodaltons, and then we're going to measure, or we're going to try to assign MBP only using the HNCA experiment. Now, here is a primary sequence of MBP, and we have color-coded each of the amino acid by the number of matches it would have in an HNCA spectra. Essentially, if I'm sitting on the HNCA, looking for my C alpha minus 1, and then count the number of matches that you would have. Now, if you take an HNCA where the resolution in the carbon dimension is about 42 hertz, you can see that most of the amino acids have about closer to 15 to 20 matches. They're highly red. We are able to uniquely assign only very few amino acids. Now, if you take the same spectra and then the sample made in pyruvate, and if you're able to increase the resolution to about 4.8 hertz, now you can see that the picture is very different here. You can see we have a lot more unique matches that are in green. And now, even for the other residues that are not uniquely assigned, we can see the number of matches drops down from 20 to 2 to 5. Now, this is only using the position of the central frequency. What if we start using the line shape as well? So here's an example. I'm sitting on a particular residue 259, and I'm looking for a sequential assignment. Now you can see that there are two resonances that have the same central frequency, but only one of them matches the side lobes, whereas the other one does not match the side lobe. So we can simply take a correlation between the different matches, and using that correlation, if you assign them, we can clearly distinguish as to what is a match and what is not a match. Now, using that, we are able to get both the central frequency as well as the line shape. We're able to get about 98% of MBP assigned. There are still some problems over here where there's still some degeneracies even with this matching, but we're able to get a vast majority of them assigned from a single HNCA spectra. Now, again, here, just to summarize here, we have two important properties. One is a high resolution central peak that's devoid of coupling. And then you have the coupling here, which gives us a side lobes, which we call the residual scalar coupling. And this can be used to match things where we have ambiguity. And here I show you the different occurrences of valine within the same protein, and they all look very similar with the C2, with the coupled to the uncoupled ratio being very similar across all the valines. Now, there are some practical considerations while using such a method to do your assignment. You need to acquire long acquisition times to get the resolution, and that would mean about 60 to 90 milliseconds. And we normally use non-uniform sampling with about 8 to 10% sampling of the entire three-dimensional grid. Deuteration is mandatory because without deuteration, your carbon will relax so fast that you're not able to get that desired resolution. And deuteration is pretty simple in this case because you don't need to buy deuterated pyruvate. You can actually get the protonated pyruvate, and then this position is acidic. So all you need to do is that take it in a liter of D2O and then decrease the pH to about 13, let it sit for about 40 minutes, and then you can add the phosphates to bring the pH back, and that's hydrogen is exchanged to deuterium. And one other important thing you need to do is that when you're doing this exchange, you got to do it in a sufficiently dilute condition, because if you're concentrated, you'll get aldol condensation, and you want to avoid that. So with these simple techniques, it's similar to growing your regular bacteria, but instead of using glucose, you're going to be using pyruvate, and you can get these beautiful peak shapes. Now, I think I have a few more minutes left. So I want to switch to a different topic and come back to how we can connect the two parts that I'm going to say is, can we achieve these amino acid specific peak shapes using pulses in a sample, let's say, that's grown on glucose? So the idea is that in your HNCA experiment, and why I'm in love with the HNCA experiment is that because in your HNCA experiment, the carbon magnetization is transfers only during the encoding time period, which means you can use the entire relaxation of the carbon magnetization to encode resolution into your carbon spectra. But as you know, the carbon is connected to two other carbons, the carbonyl carbon, which can be decoupled, but then you have a C beta. Now we want to design pulses that can effectively decouple both the C beta as well as the carbonyl carbon without causing any Bloch-Seeger shifts. So for this, with the help of Paul, we designed some pulses using optimal control theory. And with the want of time, I'm going to show you some results rather than going to the details of the pulse. So now here you see an HNCA spectrum along the carbon dimension. You can see the C alpha, C beta splitting. And now we are applying a C beta pulse, a C beta refocusing pulse right in the middle of the C alpha evolution. And you can see that being decomposed into a singlet. And you can see the singlet occurs right in the middle of the spectra without causing any Bloch-Seeger shifts. Here we are using the pulse just only once during the spectrum. So earlier we published these decoupling pulses in 3 different varieties. We call them BATCOP1, BATCOP2, and BATCOP3. As you can see, BATCOP1 has a decoupling profile that starts at about 10 ppm C beta and ends at about 32 ppm, which means it will decouple all these amino acids, but it will not decouple the amino acids that's shown over here to the left. Whereas BATCOP3 will decouple all the amino acid except for serine and threonine, which have the C beta on the other side. And BATCOP1 will decouple only things that are in this particular trough. So by running 3 different HNCA experiments, we can get an on-off switch for these amino acids. One of them will have one of them decoupled, and the other will have a larger set that's decoupled. Now
2 minutes, Hari, 2 minutes.
Yep, that'll be more than what I need. So now we are switching to what we call a gradient decoupling method. So instead of having a yes-no decoupling, we are offering a pulse along the C beta frequency that has a profile like this. So for example, in the blue HNCA experiment, a C beta, a C alpha, whose C beta frequency is here, will be perfectly decoupled. Whereas for that same amino acid, in the green experiment, it'll not be decoupled. In actuality, our pulses look something like this, where you get a profile of fully decoupled, partially decoupled, or fully coupled spectra. Now, we run about 4 different HNCAs with different decoupling pulses, and this pulse are just a single pulse in the middle of the C alpha evolution in your traditional HNCA experiments. And depending on your C beta frequencies, your C alphas will either be fully coupled, partially coupled, or fully decoupled. Now, using this, and also combining this with the CO frequency, because the C alpha is also connected to CO, we can get intricate line shapes. And using these intricate line shapes, what we can do is that we can not only use them for assignments, we can also triangulate the frequency of your carbonyl and the C beta without ever having C beta or the carbonyl transfers. And this is going to be important for larger proteins when you go to higher magnetic field strengths. So here I show you the correlation of the C beta from experimental methods on the y-axis and from a triangulation without ever touching a carbonyl on the transverse plane. And here the same thing you see for the C beta from a triangulation from 4 different HNCA experiments to the C beta that we observe experimentally. So these can not only be used for assignments, they can also be used to get the frequency of the passive spins. They also work in proteins that are larger. Here you can see the different peak shape that are able to get at for MVP. And here are proteins, real life proteins that are at about 150 micromolar. And they also work in Lewis Gay's favorite, the proteasome. So you can see the peak shapes. And here, what we do is that we take the C alpha, and while the C alpha is evolving, we offer a single pulse right in the middle of it. And this pulse creates the C beta specific peak shapes and also decouples the carbonyl. And we don't do this twice to compensate for Bloch-Seeger shifts because we don't have any Bloch-Seeger shifts. We call these pulses beluga. And these whales, other than being cute, they also use echolocation. And we thought that will be an appropriate name for it. Again, the idea is not terribly new. Similar ideas were thought earlier by Roland Rieck. And what we're doing is that we're combining those ideas with novel pulse design to get them. So I've showed you 2 different techniques. One using biochemistry, where we use the pyruvate and the way the pyruvate gets incorporated into different amino acids to create peak shapes. And the other is using optimal control theory. these two are not orthogonal, I can take a pyruvate sample and then use these pulses to further get delineation where we have batches. So again, this is a new way of using spectroscopy and biochemistry to get amino acid specific peak shapes and then use them in assignment and also to get information. What we're doing right now is we're working with Ilya Kuprov to use this in machine learning, because you not only have these peak shape, but you also have coupling constants that are embedded in these peak shapes, which you can also use to get information about secondary structures, which can also be used in the assignment procedure. With that, I'd like to thank the people who did the work. The optimal control design was done by Paul Coote. The pyruvate labeling was done initially by Scott, followed by Abhinav, David, and Andras. We are trying to incorporate our pulses into the standard Bruker library. The BATCOP pulses are already available, and all you can do is do a get prosol, and you'll be able to execute that with the help of Wolfgang. And our ability to analyze these spectra and peak shapes, we are working with CCP NMR to incorporate this into the standard CCPN package. And then Ilya and I are working on machine learning approaches to make this automated. And with that, I'd like to thank you, and I'll be happy to answer any questions.
Thank you, Hari. That was an excellent talk. I don't see any questions, but I had a question that's related to your pyruvate work. These peak shapes that you get of the non-coupled and residual coupled line shapes, will they be different with different strains of E. coli that you might use? And could this method be used, say, with using yeast?
Great question. We haven't tested different strains. We have tested BL21D3 and P lysase. They seem to have the same pattern. We have tested different induction times. They don't seem to change, and temperatures, they don't seem to change. But other than that, I haven't tried other strains of E. coli. And yeast, I would never know what to do with that.
Okay, I think that'd be interesting to try it on a eukaryotic system. And one other question is, what do you think?
In the eukaryotic system, some amino acids needs to be provided, right? Is that right? I don't know much about yeast.
Well, yeast can be grown on minimal media.
Okay.
So that might be of interest. Also,
is deuteration
minimum
possible in yeast?
Yeah, that's the question I have. Could you do this in yeast?
Is deuteration possible in yeast?
Yes.
Okay, then we could try it in yeast.
Okay. Any field strength requirements? I mean, could you do this on a 600 or a 700, or do you need a higher powered magnet?
Nope, 600 or 700 would be good.
Okay, great.
That's less, as long as we can get the resolution.
Okay, and I think there might be a question. Oh, we had a comment. Very cool, Hari. Thank you. So anyway, thank you, Hari, for a most excellent talk, and I'm glad that we had you before Lewis. That would have been a shame if you were after Lewis and you just sort of gave up. So it's time to introduce our